broadcast ourselves on YouTube. Just we are streaming these to YouTube, but we're not answering any questions to YouTube. Okay, so welcome to YouTube as well. As I was just saying to those of us in the Zoom room, I am going to be broadcasting this to YouTube. I'll be answering some questions for the next 10 minutes here, but when we begin at past, I'm only going to be answering questions from students in our Zoom room. And if you want to be one of those people asking questions and getting your questions answered, then go over to the TT website, tailoredtutors.co.uk, and join the free trial there. And then you can follow the links to the live classes for biology. And that's exactly where we're going to be. Today, we're going to be looking at some practicing of the exam technique that I went through with you last week. So I did 3 live classes last week, one on question analysis, majorly important in A-level biology, then answering technique, also majorly important, and then looked at some exam skills. And this week, we're going to be practicing those things to make sure that you are implementing them in your revision so that when you come to do your tests in a couple of weeks, then basically they're going to become a bit more ingrained in you, a bit more second nature, and then you're going to start picking up those extra marks and extra grades, which is what it's all about. Hi, Ahmed. Hi, Sophia. So you pull up the chat. Ahmed's already done that. He's already changed his settings to panelists and attendees. So thanks for that. Okay, so I'm going to be giving you a little bit of homework this week. It is very minimal. Your homework for today is going to be to send me some application questions. Now, this isn't really a sort of an official term application questions. I suppose AO2 and AO3 are the exam speak jargon for that. But you tell me, I get asked to do application questions the whole time. So I want you to send me either a screenshot of the application question you're struggling with, or to just let me know the exact exam code. A screenshot's probably going to be easier because students will often not send me the right information. They'll say it's paper 1 from 2015, question 6, but then I don't know if it's an AS paper or an A-level paper. I also need to know the exam board. So let me know those bits of information. If you are going to send me the code, I need to know the exam board, AS or A-level paper, what year it is from, and what question number it is. If it's an old spec question, I will also need to know the unit because they had separate unit exams back then. All right. On YouTube, why not macromolecules as well? Give me a bit more context for that, Astan. I don't know what you mean. Okay, how long else have we got? Yeah, I'll pull up my iPad in a moment and go through some bits and pieces. I've got a few little sort of handouts that might be useful, sort of side handouts I'm mostly going to be getting you guys to do the work today. And I'm going to basically just put up a question and get you guys to do it more or less without any of my help. And then we're going to reflect back on it and see what clues were there to help us get more marks or ideally full marks. Does anyone have any questions for me before we kind of get going? My hands are super dry. Moisturize them up a little bit. What else has been going on inside TT recently? We've just updated all of our content guides and exam guides. So if you are on the YouTube channel and maybe you tried TT a couple of months ago, maybe in September at the start of the year, things have changed quite a lot since then. So I'd highly recommend checking out that free trial, just going through. And I mean, normally you only use the free trial once, but I don't remember the settings off the top of my head, but I think after 3 months has gone by, you can do a free trial again. Is lipid a macromolecule? Interesting question. It's not a polymer. Is it a macromolecule? It kind of depends, I would suppose. I don't know the technical definition of macromolecule, like large molecule. Those fatty acid chains, which obviously a triglyceride is, there's no limit to how long they could be. There could literally be 10 million carbon atoms in a row, in which case I would say, yeah, it's a macromolecule, but you're not going to find those in biological systems. So it's not a polymer because it doesn't repeat the definition of poly and mer, many units. So is it a macromolecule? To be honest, I would go straight to Google and ask that question. It's not a question that your exam board is going to ask you. So that's why I don't know it. Well, it's all been very nice and relaxed here. Well, what can I get you doing in the meantime? Get yourself, make sure you've got pen and paper, because it was going to be probably handy to be answering these things on pen and paper. You're going to need a calculator. There is a couple of little math skills questions in here, but not, I don't want it to be the major focus. The best way of getting up to speed with the math skills is to find which ones you are having trouble with. There are quite a few. The really common ones, obviously, are percentages. We've got ratios. We've got the stats questions fall under the maths part of the specification. And they are routinely asked. So making sure that if you do, some people really struggle with the math skills, and I get it. I'm definitely not a natural when it comes to maths, but if you do lots of practice, then they become basically an algorithm. I do this first, like percentage change. I do the big number minus the small number to find the difference. And if I'm doing a percentage increase, then I divide by the small number times by 100. If I'm doing a percentage decrease, I divide by the bigger number, divide by 100. So it just becomes this algorithm. And then the other skills involved are with biology questions, you won't get in maths questions, is like, which numbers do I need to extract from the question stem? And that comes back to our question analysis session that we did the other day, like which underlining the bits as you're reading the question so that you can go back to the numbers very easily, just paying attention to the units. Is one of them per 15 minutes and one of them per hour? Is one per day and one per week? Obviously, there's a mixture. Megan in the chat, exciting news. I got an A in your mock yesterday. I'm assuming this is contributing to your grade. Do you know if it was one of the ones that is going to be one of the contributing factors? Every school and college is doing it differently this year. I've been talking to a few heads of department. A lot of them are using about 5 different assessments. Thank you, Megan. That's so good to hear. I'd love, Megan's one of our brand ambassadors. She's been using TT for at least a year now, Megan, if not longer than that, because you started in your AS year. Like, it's probably been a gradual process reaching that. What would you say have been the headline things that have made the difference to sort of improving the grade? You were getting solid Ds and Es in year 12. So any year 12 student watching this right now, it totally does work. Megan has done a lot of hard work. She's saying 100% wouldn't have done it without TT. However, like, we can only show you the path. You are the one that has to walk along it and do the hard yards. So, okay, TT method, going over every mark you get wrong and then practice. I mean, literally, it sounds, I've been a broken record this week. We've had a whole lot of students using the course out for the first time over Easter, and all are saying, like, how do I revise? And I've just been more or less on loop saying TT revision method, TT revision method, TT revision method. I sound a bit like, yeah, like a broken record, but it is the only way you're going to see your errors and then improve. Like the math skills, perfect example. If you are shocking at uncertainty and measurement questions, like they're difficult, it's one of the more difficult math skills, then the only way to get better is to practice. There's not that much to know. It's all about practice when it comes to those things. Hi, Kia. Okay, so interesting question from Scarlett. We've got a couple of minutes before we really get stuck into it. What would I recommend year 12s doing during this time? Go over content still or past papers? So I'm going to give you 2 scenarios. One is you haven't yet completed all of the AS content at school or college. I think if that's the case, your first priority is to complete the content. So using TT, for example, to get ahead of school or college, if there's still a module of AS content left to do, getting to the point where you have completed all of the AS papers so that you can begin the TT revision method using the AS papers is going to be the best way of going about that. If you have more or less finished the AS content, and when I say finish the content, I don't mean that you know it perfectly. Perfect knowledge basically doesn't exist in biology. So as soon as you've got one exposure to each of the topics, that's when you'd start doing TT revision methods using the past papers. And then that's going to throw up. It's going to shed light on the fact that, oh, this topic over here, transcription translation, I'm terrible at that one. I'll go and point some more revision and top up my transcription and translation revision. So I would say Scarlett, where are you at? I'll ask you, where have you completed the content for the AS yet? What else would I say about that? There's a document. I wonder if I can find it in the next 2 minutes here. I probably can. So let's. Oh, actually, I've probably got it. I might have a link direct to it here. Nope. Let's see if I can find this in the next. Oh, it's the study planner. I wonder if I can. I just have to re-upload it. PDF. Let's try this. Yeah, module 1 for OCR is like not the, it's all the practical skill stuff. So you're going to get lots of exposure to that over time. So I wouldn't worry if you haven't completed module 1, because that it's going to happen over time. Okay, so let me just share my screen before we jump into the full live lesson. This is like our sort of, it's still a work in progress, to be perfectly honest with you, this here document, but I think it's going to be useful to you right now. I'm going to skip all of the top stuff. I often get asked how much time should I spend per subject per week. It's going to be, in fact, roughly 4 hours of classroom time, roughly 4 hours of homework time, and roughly 4 hours on top of that. It gives you about 12 hours per subject per week in normal times, which gives you 3 subjects. It's like 36 hours per week, which is the full-time course that A-level should be. So I want to just draw your attention up here. I will share this link with you. I'm going to add a lesson to the Common Room course that gives this in here. So in year 12, we can match the colors. So we got learning new content. Obviously, the bulk of our time, 3/4 roughly here, is learning new content. And this is some example activities of how to do that. And then revising past content is this darker yellow orangey here. And you can see up till Christmas, it's basically mostly learning new content with a little bit of revising past content. It's really important to build a routine from the beginning that incorporates revising past content. Otherwise, if you just do new content, new content, new content, you do all of this, by the time you get to March and April, then you probably don't remember very much of what you did in September. So it's really important to do this sort of spiraling, looping, revising of past content. This is mostly going to be topic-based question packs and stuff. And then the reason why I gave you 2 answers, Scarlett, is because we're here, right? And so now we're moving to the TT revision method, which is using the whole past paper. So now at this point, we can reflect back on the entire AS using the AS papers, and then we can cycle through them and learn as much as we possibly can. And then usually in June of year 12, you start looking at A-level content for the first time. And so that's where learning new content comes in again. In year 13, it's the same pattern, but we can use AS papers from the beginning. So here we can learn new content and we can revise AS content using TT revision method all the way through. And now obviously in year 13, we're now looking back at the entire content of everything and trying to bring it all together. But it's really important that we incorporate this revising past content in with our normal study throughout the year. And then obviously, as we get towards the end of the year, April, May, June, is when we do the full TT revision method. Looking back, it's the most holistic, all-encompassing revision method. What I put down here, again, this is definitely something that needs a work in progress. But let's say in week 1, we're just learning, like your first week of A-levels ever. Obviously, you can't do revision at this point. But then if you can try and stagger, so if in week 2 we're learning topic 2, let's say this is DNA, we can then revise topic 1, which might have been biological molecules. And then when we get to topic 3, which might be transcription and translation, we can then revise DNA. So you're always trying to do that active recall of something that's in the past, basically revising past content. That's what I mean by this. It definitely could be an improvement. And I will share this link with you guys now. As I said, it's going to get revised, that there. Document over time, but probably not till the summer. So that's as good as it's going to get for now. All right, well, I'm already 3 minutes beyond my starting schedule. I'm just going to check in with YouTube one more time just to see if there's any questions in there. I've just been doing mass transport with TT. Excellent. Okay, cool. Right then, let's get rid of some of my unnecessary windows here, and let's jump in straight away to the old iPad. Okay, so what I've got going on here, I'm going to minimize that, bring that across there. Thank you for pausing my screen share Zoom. Okay, so I'll bring my chat in so I can see it a bit more front and center. Okay, so what this is sort of a bit of a plan for the week. Today is Tuesday. We've got exam technique practice. So we're practicing some of the skills that we picked up yesterday, drilling them, trying to improve them. There's kind of a random selection of questions that I just pulled to hopefully highlight some of the things we've been looking at. Your homework today. I want to know what, okay, first question to the chat. What do you mean when you say application questions? That's my question to you. I'm going to give you 30 seconds to answer that. And are there any application questions that you can remember that you were like, oh yeah, that was a really tough one? Or if you can describe them to me, I'm probably going to know them. Emily, so you can't even work out where the topic comes from. Questions that cannot be simply answered by topic knowledge. Definitely, Sarah. Question doesn't explicitly mention the topic or keyword, so you have to like deduce it or infer it. Yeah. Long worded questions that aren't just recall from Emily. 100%. So yeah, it's like topic which isn't directly content knowledge. You have to apply your knowledge to a new situation, like draw the links, apply content you know to something you don't know about. Yeah, absolutely. Using multiple different parts of the course to figure something out. Yeah, exactly. So this is how it moves across the membrane here. Maybe this can't happen because it's too large to cross the membrane. If that doesn't arrive, then this reaction doesn't happen. If that reaction doesn't happen, what's the end result? So being able to work out systems forwards and backwards, respiration or photosynthesis is a good one for that. What happens? This molecule prevents this thing happening. If this thing doesn't happen, you've kind of got to go all the way back up the chain. Where does that have its impact? Okay, Emily. Okay, so you've given me an example question there. I'm just going to stick that on my notepad so I can keep it for later. I send more messages to myself than anyone else ever sends me. Questions that link topics but not directly, so you have to apply a long passage of text. Okay, so I've got a nice idea. So any that you come across before tomorrow's live class, take a screenshot, let me know the exact question code, send it to me, and I'll pull some of them together and we'll see if we can break some of them open tomorrow. So tomorrow's going to be mostly application questions. So tomorrow's live class on application questions. I'll find some myself, but I really want you guys, I would like everyone to send me one, or at least to look for one. Then your homework tomorrow, I'm going to put together a little mini mock for you. It'll be 2 to 3 AS questions, 2 to 3 A-level questions. Send them to me in a comment in the course, Sarah. And then, so that's going to be your homework. Should be about 45 minutes to an hour, depending on how many questions are in there. Then we're going to look at that mini mock and see again how we can break down those questions. And then your homework for Thursday is relax for a little bit. And beyond Thursday, TT revision method is going to be what's going to serve us the best. You've not missed a lot, to be honest, Lucy. I've just gone over the plan of what we're doing. So we are about to get stuck right in. Okay, I'm going to give you, what am I going to give you? Because we've got 3, 4, 5, 6, 7, 8 marks. I'm going to just do part 1 first of all. So we're only looking over here. And I'm going to give you, this is a weird one. I'm deliberately not helping you with this. I'm going to do my analysis after the fact. I'm going to give you 1 minute for this first question. Super tough, rude awakening. You've got 1 minute. The timer has begun. Let's go. Describe and explain the appearance of one of the chromosomes in cell X. Obviously, let's read this bit as well because that might be important. Again, feel free to either do this in the chat or write this down on paper because I'm going to be working on exam technique. It might involve revisiting your answer. So if you do have pen and paper there, it's probably a good way to go. 25 seconds. This one is like rapid fire. You're not going to get any freebies today. They're all going to be tough ones. All going to be tough time limits anyway. Okay. Well, I'm going to just analyze it. You might be finishing off your answer. Summarizes the process of meiosis. The circles represent cells and the structures within represent chromosomes. Okay, nothing too much to go through there. So we've got describe and explain for 3 marks. That's a command word of marks. The appearance of one of the chromosomes in cell X. So we're looking at this guy. So like, if I were to reword this question, the appearance of or the behavior of chromosomes, the appearance of, what do we think this means? The appearance of. Condensed, yeah. But why do we need to explain its appearance? It's not normal, right? It doesn't normally look like this. We're going through meiosis. It's condensed. Very nice, Fatima. So why is it not? How is it different to how it sort of normally is during interphase, I suppose, or early interphase, if we're being precise? So we don't have to do both. So we only need to describe one. So basically, it's describe it. And maybe the explain is like, it's not a great explain command, use a command word, but like explain, or why is it different to how it normally is? Okay, so again, if did anyone, my question to you, did you apply carbs? Did you run through the bullet points of key terms before you started writing? Yes or no in the chat? I was very harsh on time. I maybe could have given you a little bit more. Okay, so maybe that's my, maybe next time I'm going to give you 1 minute for for prep, no writing, and then 1 minute for writing. Maybe that way is going to help sort of. Okay, so a little bit of key terms searching. If we're going to just do a quick blurb, just the first things that come to mind, relevant key terms for this question, the appearance of these chromosomes here. Condensed. First 3 out the blocks, condensed. Visible, centromere, sister chromatids, DNA replicated. What's joining them together? Centromere. I would assume all of these things are going to get the answers. Okay, so now I'm going to reveal the answer. Oh, why have you stuck yourself together? It's a bit weird. Let's get rid of you. Let's switch to red for a little bit of. Chromosomes formed of 2 chromatids. So we've got chromatid 1, chromatid 2. These are sister chromatids because they're joined by the centromere. If I were to draw these guys, these would be the blue one here, and the red one here would be non-sister chromatids, if we're getting fancy about it, assuming they're a homologous pair, which it looks like they are. Because DNA replication has occurred, so why are there 2? And this is what it would normally look like if it was condensed. However, we've got 2, so where did the other half come from? It came from DNA replication, and the sister chromatids held together by a centromere. Key term, chromatids, DNA replication, centromere. Alex got 2, missed the obvious for DNA replication. Okay, so what stage of carbs could possibly have helped you with that? You know, look at that B. It looks like a D. What stage of our question answering method carbs could possibly help you catch the obvious? What do we do in self-check? What's the question you can ask yourself? what other key terms can I add? Anything that's relevant. I mean, if it's really fringe, it's probably not worth it. It's almost never going to deduct a mark. The only time you can ever lose marks is for what they call the list rule. If it says name 2 processes for how things cross the cell surface membrane, and it's asked for 2, specifically 2, and you give a list of 5, well, let's say 6, 3 of which are right, 3 of which are wrong, you get 0, because you can't just give an infinite list when it's given you an exact number of things that it wants. If it says 2, you can give 2. If you write down 3, 2 of them are right, 1 of them are wrong, you get 1 mark. But apart from that, you're basically safe. As long as the key terms are relevant, put them in. All right. Describe is okay. We've got 2 marks here. I'm going to get rid of my scribbles. This should be quite easy. I'll do the first bit for us. Describe what has happened during division 1. I mean, so again, I'm going to give you 30 seconds. Why not? Let's try this. Let's try this as a format. 30 seconds for thinking. You're doing your question analysis. 30 seconds has begun. So no writing right now. 30 seconds. You're probably doing this bit of carbs, I hope. Okay, so that was just our thinking time. I'm going to give you another 30 seconds for 2 to 3 short, concise bullet points. Again, you can write these down, and I'm okay, and I'm going to give you another 30 seconds for finishing off anything you've done and the self-check. Can you add any more key terms? Okay. Describe what has happened during division 1. So we've got Shabnam, telophase 1, splitting the 2 homologous chromosomes into different cells. There is a what phase is happening here? I suppose telophase. Yeah, what we've got this one here and this one here. So let's see if we can get some more key terms. Oh, you've got 2 homologous chromosomes. 2 homologous chromosomes. So this is an example of where the devil is in the detail. Yes, I think I'm actually going to allow that. I think that's going to be fine. Cytokinesis, physical division of cell, half the DNA into each new cell, full set of chromosomes. Yeah, again, homologous pair. I think we can definitely get the word homologous pair. Each homologous pair has gone into each new cell. Independent segregation in meiosis 1. Homologous pairs of chromosomes separated into 2 daughter cells. So I think homologous pair, because these look to be a pair, with 1 coming in here and 1 coming in here. 1 of each daughter cell, 1 into each daughter cell, or it gives us the haploid number. Berry, I've seen your question. Chuck it in the Q&A and I'll come to it maybe at the end, or like when we're at a suitable break. And obviously ask me questions on the course if you have questions like that. So this is just a point fact. Identify, quite a rare command word, but find one event that took place during Division 2, but not during Division 1. I'm assuming that must mean. I think we're just missing the number 1 off the end there. Can only be that, I assume. So what does happen here that doesn't happen here? Tia, separation of the sister chromatids. More key terms, more key terms. What has happened to separate the chromatids? I love your short sentences. This is excellent exam technique. But now second sentence, or you can put a sentence in before or after. How do they separate? What was joining them? We can call this independent segregation. Okay, so spindle fibers pull sister chromatids apart from their centromere. The centromere splits or divides or is broken. The chromatids are separated. Okay, so they will allow either here, separation of sister chromatids, or division. This is a slash means or division of centromere. When we're doing practice papers, always try and get them all. And another bit of technique here, if you wrote this one and you get the mark, well done. You got 1 out of 100%. But you didn't think of writing this, then in green, you should that in. Centromere splits or divides. Because next time it could say, other than the separation of the sister chromatids, what action took place here? They next time, this might be the only acceptable answer. So knowing or being able to access all of the correct answers gives us more options when we're moving on. And then absolute classic, I'm not going to dwell on this, but name 2 ways in which meiosis produces genetic variation for 2 marks. Boom, boom. Go, go. Alex, straight in there, crossing over an independent assortment. You can also say independent segregation. Just the difference between assortment is in metaphase when our pairs line up on the equator. When they're sorting themselves out, if I just call this one A and this one B, obviously which way around they go, A or B, maybe this one is B and this one is A. In fact, maybe mother and father is a better way of doing it. Let's go father, mother. This one we can have mother and father. And this one we can have, what are we doing here? So it's obviously random which maternal or paternal chromosomes goes on the left or on the right. So when they're assorting themselves in metaphase, then that's the assortment. And then the segregation is when they're being segregated or when they're actually being separated or pulled apart during anaphase. So technically, I suppose I could write that out. We can do assortment is in metaphase and the segregation is in anaphase. And that's when they're being pulled apart like this. Could you say formation of the chiasmata or chiasma? No, because the question here is what is happening in 2 that is not happening in Division 1. So it would be the other way around. The crossing over takes place in Division 1, but not in Division 2. If the question was reversed, then yes, but not for this question. I'm pretty sure we're going to see exactly that on the mark scheme. There we go. Sticking on a theme here, I'm not going to dwell on this one too much. fastest finger first to this one up here. Which letter represents the statement chromosomes line up on the equator? I literally just gave you the answer to this one. Emily was in there first. Anaphase, interphase, metaphase, prophase, telophase. Let's not dwell on it. So simple as they come, that one. That is absolutely metaphase. Metaphase for the middle. All right. I'm going to read through it so we're all on the same page, and then I'm going to give you the questions. Scientists looking for treatments for cancer are investigating the use of substances called kinesin inhibitors. So the treatment for cancer. So now I'm thinking, what's the disease of cancer? Uncontrolled cell division, when mitosis runs out of control. So that's like, okay, so it's got something to do with cell division. Kinesin inhibitors, or KI. These inhibitors prevent successful mitosis. Okay, wonderful. It's told us that. Some kinesin inhibitors cause the development of a monopolar, some, but not all, just might be relevant here, cause a monopolar spindle. Figure 6 shows the chromosome attached in a normal mitotic spindle to a monopolar mitotic spindle. Okay, so we've got normal, the centromere attached to the pole, and a monopolar mitotic spindle. They're all coming in the same direction. Suggest why the development of a monopolar mitotic spindle would prevent successful mitosis. I'm just going to let you have this one. Okay, I'm going to give you 30 seconds for the thinking. I'm going to give you 1 minute for your writing and 30 seconds for your self-check. So your 30 seconds of your question analysis, 30 seconds starts now. Okay, so I'm going to give you 1 minute 30 because it's easier for me to change that on the timer. 1 minute 30 for your bullet pointing. In fact, you probably don't even need a minute 30. I'm going to give you a minute and then 30 seconds for self-check. Okay, so 30 seconds for that self-check can start now. Jasmina, I'm pretty sure you're AQA, right? Jasmine, let me know your exam board. I'll check the link in your version of the course. Clearly the link's working for most people here. Okay, time's up. So what have we got? Chromosomes on, like, really nice short use of sentences there, Megan. So chromosomes on one side only. We could maybe say, we're not talking about homologous pairs here, potentially. Yeah, so after cytokinesis, one cell has all the DNA, the other has no DNA. The cell's not identical. They're unable to carry out their growth and function properly. Chromosomes do not split at the centromeres. Okay, let's pull up the mark scheme whilst I just... So these suggest questions are not expecting you to know anything out of the ordinary. Okay, so we've got no separation of chromatids, chromosomes, or centromeres for 1 mark. I think pretty much everyone that sent me their answer got that. All the chromosomes or all the chromatids go to 1 pole. Double chromosomes doubles the chromosome number in 1 cell, and 1 daughter cell gets no chromosomes. You could say anaphase is prevented. You can, if you know the term chromosome non-disjunction, you're allowed to say that there. Except DNA for chromosomes and except ploidy or polyploidy. 1 end cell ends up having multiple versions of the chromosomes, and one ends up with basically none. Oh, good spot, Megan. Yeah, there are. Maybe I should just remove the button for the week 1. The live class links are changing right now. I'm pretty sure. I'm just going to do a quick poll just to make sure that I have got the link right in all your courses. I'm pretty sure I copy pasted it. So in theory, if it's right in one place, it should be right in all the places. And Alex is here, so I know that, yeah, okay. So I've seen enough to know that the links are all right. Okay, excellent. Let's find one we can do a bit more in. This was a bit of an interesting suggest one. Okay, let's go through. I think we have time. Where are we at? We're on the hour. Make sure you're using the link for the week 2, not the week 1. Last week's link was a different link. So it should all be working for you in the one next to today's date. All right, okay, so where have we got? Figure 1 shows the structure of a fat substitute. We have, okay, so what's this similar to? So these are both suggest questions here. Suggest questions basically mean, what is it similar to? So what does this molecule kind of look like? What is it structured like? It looks like a triglyceride, right? So in a triglyceride, we have glycerol. And then we have our 3 ester bonds. And then, well, I don't know why I've drawn another circle there. Then we have our fatty acids. So this fat substitute cannot be digested by the gut lipase. Suggest why. So let's go through carbs on this. Command word is suggest. We have 2 marks. What's the actual topic of the question here? Excellent. Nobody's giving me lipids or triglycerides as the topic of the question here. This is an enzymes question. We're specifically talking about why can an enzyme not break down some molecules? It's kind of the opposite of why are enzymes specific to one substrate. Okay, so then quick key term, refresh, active site, tertiary structure, enzyme substrate complex. Well, we've probably got them in those 3, right? But I mean, we can talk about it's a protein, it's complementary, not complementary, if the case may be, because we're talking about not being able to hydrolyze things here. So this is a kind of a nice suggest question. It doesn't expect you to know anything about this specific molecule here. It's just asking us to apply our knowledge of enzymes. So we've got different or wrong shape or not complementary. Obviously, this is probably our nicest language, our best language to use. You could say that the bond between glycerol and the fatty acid is different to the one between propylene glycol. And then the second point, unable to bind to fit to active site, no enzyme substrate complex formed. What is it similar to for these suggest questions? Okay, next up. I got asked about this in the course this week, and it's what brought this question to my attention. This fat substitute is a lipid. Despite it being a lipid, it cannot cross the cell surface membrane of the cells lining the gut, but any cell surface membrane, doesn't matter which one. Suggest, which is our command word, why it. So again, we can try and replace these things. Cannot cross the cell surface membrane. Let's rephrase this question. Well, okay, let's go through carbs. Fatima, can you write ES complex? Technically, no. According to the specifications, you're not supposed to. This year, your teacher is marking it. So this year, I would say you can write ES complex safely because your teacher will know what you mean. Okay, so carbs, actual topic of the question here. What's the actual topic? We've done our command word. We've got suggest, which means what is it similar to? Cell surface membrane. Transport across membranes. Very nice. Key terms. So we've got things not being able to cross the cell surface membrane. Give me some key terms. It's polar, charged, too large, too big. No protein channel or no carrier protein. That's pretty much it. Nice little synopsis. So let's rephrase this question. I'm going to add an extra step in here. I'd like you guys to rephrase this question for me. Just in a sentence, how would you rephrase this to make it easier to answer? What do we got? Suggest why something why could something not cross a cell surface membrane? Pretty much, Fatima. Yeah. What properties of a lipid make it impossible to freeze diffuse? Potentially, I think we can go more. It's a suggest. So again, they're not expecting you to know anything about this. You're not expected to know what this bond is here. They've not named it. This is a black box of unknown that they cannot expect you to know absolutely anything about. It's not on the spec. This bit, we know, but black box of unknown, which means you've got carte blanche. You've got absolute blank check for writing any sensible suggestion. Give a reason why a substance cannot cross a cell surface membrane by simple diffusion, I guess, because normally a lipid would go by simple diffusion. And you've all answered the question. I mean, like, so this, I got asked the question, surely this molecule isn't hydrophilic. No, it's not. Highly likely not to be hydrophilic, but you don't know what this is, right? Maybe you do, but the chances are you don't know what this is. So this could be, it's not expecting you to know. It's saying why any sensible suggestion, why any molecule is unable to cross the cell membrane by simple diffusion. And it won't accept lipid soluble because it is a lipid, and it has told you that. So the only non-acceptable answer is basically because they've told us a little bit of info in the stem here. So hydrophilic, polar, too large, too big, any sensible suggestion. So what I'm trying to do here is show you that suggest questions are not as scary as you might think they are. Really underneath it, they're not expecting you to know anything. They're not expecting magic from you. It's always, what is it similar to? What do I know? Because the answer will be, or should be, within the realms of something that you know. Okay, how are we feeling about suggest questions? Any change in your mood when it comes to suggest questions? More confident, not a fan, but better? And you won't get them all. I'm not going to pretend that every suggest question is kind of this simple. I've chosen what I would call like scaffolding questions to like help us get there. I don't get all the suggest questions, but it's not expected. Students will often say, how on earth could I be expected to know that? Well, like this isn't hydrophilic. This molecule isn't. I'm pretty sure it's not glycol. I don't know exactly what, but propylene definitely isn't hydrophilic. But it's not saying, tell me about this molecule. It's telling me, tell me about the principles of cell surface membranes and what goes across them and why. So that's where we're at with that. Okay, changing topic. These are all suitable to all exam boards. They're mostly AS topics, but I'm not really going into the content here, because again, I want to just bring us back to our skills. It's a couple of interesting. We've got an outline as our command word and a suggest here. Okay, so let's do all of this side of the page. We'll come on to the math skills and stuff in a second. So we've got A, B, and C. We've got 5 marks. Okay, outline is like, it's like describe, but you don't have to describe in full detail. It's like the zoomed out, in this case, properties. Do you think you need to give reasons for outline? It means like give an overview. Yeah, briefly describe, top level detail. If I said describe aerobic respiration, it'd be like glycolysis, link reaction, Krebs cycle, oxidative phosphorylation. Whereas I'm not looking for glucose is phosphorylated by ATP to produce hexospisphosphate, and that then breaks down into 2 molecules of this, that, and the other. It's kind of state, but it's like you don't have to go into the nitty-gritty detail. So in this case, it would be state or name or give properties of water which make it an ideal habitat. That make it an ideal habitat. Oh, they don't make these things easy. That make it. Do we have to say why it makes it an ideal habitat? It definitely is not going to hurt, but it will waste time. Which make it an ideal habitat for an amphibian. Yeah, I think able to dissolve solvents, probably that's good enough, I would think. All right, okay, I'm going to give you 5 marks. I would like, okay, I'm going to give you 4 minutes for answering, and I'm going to give you 1 minute for self-check. So you're going to have 5 minutes, but I'm going to break it down into 2. Let's go back to 0 seconds on that. 4 minutes starts now. Have a crack. I don't want to give you the answers. It's best to have a go at the answers, and then I'm going to help with them afterwards. I'm going to go fix my links or just delete some buttons. Actually, maybe. 30 seconds. Whoa. Okay, then 1 minute for self-check. So can you add any more key terms? It's going to give me just enough time to fix this, I hope, because I just broke it all. Ten seconds. Oh, look at that, perfect timing for me. Okay, let's have a look at this here. Name the genus of the Titicaca water frog. Well, we have the binomial naming system. You're going to have to spell this one correctly because it's given to you on the page. Must be written with a capital T because it is the genus. And we're literally just copying this word down here. Okay, then we have, here we've got the outline, which make it an ideal. For me, there should not be reasons on here. There shouldn't be, because that's not what the question, it says that make it an ideal habitat, but the question is outline the properties. We shouldn't have to justify it. Good solvent, high specific heat capacity or temperature stability, or a description that describes the temperature stability. High density, so frog is buoyant or floats, or ice is less dense than water, so that it basically, yeah, we don't have to say why. I'm very happy that they didn't insist on whys there, because they shouldn't have done, given that the command word was just, so it's like just statements. Allow has oxygen dissolved in it. And then our part 3 here. Again, we have suggest why it has evolved unusually large folds in the skin. So I don't think there was any more of the question 7 that was missing here. I'm just going to go back up and see if we've got any clues up here. Let's go. I mean, it's a suggest. Again, it's like, what is, okay, like all amphibians, I was looking, I knew there was going to be more of a clue in there, and I just skipped over it. Frogs absorb oxygen through the skin as well as their lungs. So this is our clue. I've been to Lake Titicaca. It's actually at high altitude. It's at probably 4,000 meters. So the air's like higher than probably any mountain. Mont Blanc is maybe 4,000. The highest mountain in Europe is like 4,800 or something. So it's like nearly as high as the top of the highest mountain in Europe. And that's where the lake is at. So the clue here is linked to the oxygen absorption. So any kind of folding, in fact, I saw someone had used this term on YouTube earlier, invagination, means inward folding. We see it with microvilli. which we touched upon in, is going to do what? It's going to increase the surface area of the skin, which will, or you could say increase the surface area to volume ratio. Both of those would be relevant here, which increases the amount of oxygen absorbed. It says oxygen levels in the lake are low. Interesting. I would probably link this to aerobic respiration. More oxygen to diffuse through skin is allowed. And we've got 2 to get. Any questions on that? Again, if we struggle, okay, I'm going to ask the poll here. It's probably a good time to be asking this poll question. Okay, so I want to, those who are on YouTube, I've launched a poll in Zoom. Reasons why we drop marks, and you can select all that apply. We've got FFS, which is silly mistakes. We've got analysis. This is for all 3 of these questions, and for every mark you drop, you can put the tally in there. Analysis, you didn't quite fully understand what the question was asking. We've got content knowledge. Simply you were missing content knowledge from your head and that you just didn't know. Technique, you knew the marking points. Now that you look at them, you know that surface area to volume ratio is important in gas exchange. You know that the bigger the surface area, the more oxygen would. So like knowing more stuff's not going to help you, that the technique that you adopt wasn't quite right, or possibly the skill you didn't know or couldn't do a skill. Okay, so most of you have voted there, 68%. So I'm just going to share that with everyone in Zoom. So answering technique, knowing the facts, but not using the right key terms or not writing the right ones down is 53%. Silly mistakes. I'd like to know what those silly mistakes were. I'm imagining a lot of what you're considering silly mistakes probably maybe answering techniques if you didn't use the right key term. I personally call it answering technique. Doesn't really matter. You can call it an FFS if you want to. Second, 47% was analysis. Didn't fully understand what the question was asking. Why is it important that water is a good solvent here? It's a good question. I mean, it certainly wouldn't have been first on my list, that's for sure. Oxygen, but I mean, like, really what makes water a good solvent is that it's polar, and therefore ionic compounds dissolve in it better, such as nitrates, which is much more relevant to plants, or anything with a charge, sodium chloride, what the frog is doing with any of those things, because not a huge amount of oxygen is going to dissolve in water, because the oxygen molecule is O2. It has some uneven distribution of charge, but it's not polar in the same way that a sodium ion is polar. So if it was a plant, we can definitely say ions dissolve, such as nitrates, and plants even use nitrates to make proteins in protein synthesis. It would probably allow oxygen as being something that's dissolved in it, but not very much. It's good. I mean, not a lot, really, I'd say, is the answer there. But I want to just bring your attention to, so 50% lost marks for exam technique, 47% lost marks for question analysis, only 24% of people drop marks because of content that they didn't know. So again, just making sure we're focusing our revision on the things that matter, and not, your content knowledge isn't going to get perfect. And so waiting until our content knowledge is perfect before we begin tackling answering technique and question analysis is not so good. Okay, yeah, you're quite right, Alex. It does say allow has oxygen dissolved in it. It's a solvent. I wouldn't say it's a good solvent for oxygen. Okay, where are we at? 25 past. Turning our attention over here. This is purely hypothetical. Now it's the following. It is this question continued, but cube A has 10 millimeters along each side and cube B is 4 millimeters along each side. Calculate the surface area to volume ratio of both cubes. Show your working. Give your answers to 1 decimal place. What don't you know looking at the answer right now? What do you need to know? That 2 marks. Okay. Let's calculate. I'm going to give you 2 minutes. You're going to need your calculator. Again, this might be a quick sketch, might help you visualize this. We've obviously got either 10 millimeters or we've got, how big is it, 4? "30 seconds. Steffi, you asked about latent heat vaporization. How is that an ideal habitat for an amphibian? It doesn't want the water to evaporate. Or maybe you could state that preventing evaporation is a benefit. Maybe, but I'd say less relevant. Okay. What have we got for our ratios? What's the technique here? Well, we work out the surface area of one side, so that's 10 by 10. And then how many sides has a cube got? 6 sides times 6, so that's its surface area. Its volume is 10 cubed, 10 by 10 by 10, 10 to the power 3. So we can then work out our ratios by doing surface area and volume. And we do the same technique for 4 millimeters. 4 times 4 gives us our surface area of one side, times by 6 for 6 sides, and then 4 cubed. That's our SA, and that's our vol. Okay. So again, we've got our raw numbers, 600 and 1,000 for our surface area of our big cube and our volume of our big cube. And then we have our 96. Yeah. Yeah, so this is our surface area and our volume of our smaller cube. And then we can obviously reduce these down to make them a bit more the simplest ratio that we can give. No units because it's a ratio. I'm just seeing what they've done here. As correct ratio is not to 1 decimal place. Interesting that they've reduced these down all the way a decimal to 1. What do we get here if it's my phone, not my calculator, if we express our ratios? So 1,000 Did anyone write these as 1 point, well, to 1 decimal place, it'd be 1.7 to 1 for for our surface area to volume ratio for the big square. And what's yeah, and then this one 1.5 to 1. This is an OCR one. I hadn't actually done this one in advance, so I'm just like baffled as to why they've given us our. Typically, I would choose whole numbers. Did anyone come up with this? So if you do our surface area, oh, okay, so our surface area is 600 divided by 1,000 is why they get 0.6 here, because that is our 600 divided by our 1,000 to one decimal place. So this would be 1 to 1.7 is what I meant. I wrote my ratio over here the wrong way around. And there's no additional guidance on this. Yeah. I'll come back to you on that one tomorrow because I don't have a specific answer on that. This is, I think it could well be a specimen paper. There would normally be additional guidance to like tell the examiner what to do if the ratios are not written in. See, they've given just like raw ratios like this, but the fact that it says to one decimal place makes me think there's going to be a decimal in here of some sort. So if I've just got round numbers like this, does it say express it in the simplest form? No, it says give your answers. So even if this was your number, 3 to 5 and 3 to 2, then it should be 3.0 to 5.0 and 3.0 to 2.0. So it has told us to give us, but this, as far as I'm aware, should be accepted either way around as long as your ratio is correct. So either 0.6 to 1 or 1 to 1.7. And this one, it makes sense to do this way around. Yeah, but because it says to one decimal place, you would have to write, you can't write 3 to 2. It would have to be 3.0 to 2.0 because otherwise it's not given to one decimal place. Yeah, I think this would be fine. And this one's not a problem because the surface area is larger than the volume. All right. Explain, so giving reasons why the surface area to volume ratio of an organism determines whether it needs a circulatory system. Again, for this question, I'm going to just give you 3 minutes for 3 marks. And I'm not going to give you any help apart from the help that I've given you on analyzing the question there. 30 seconds remaining. Okay. I'm going to mark it again. I'm going to ask you that same poll question. Just for the part 3 this time is where I'm going to ask you that poll question, but let's mark it first. Three marks total. Larger organisms have a smaller surface area to volume ratio. So therefore, diffusion is I would say the diffusion distance is too long. The distance between the outside, where the oxygen is, and the tissues on the inside is too long. That diffusion distance is too long. 4, sufficient delivery for enough oxygen. I would link that ideally, you don't have to in this case, to aerobic respiration. Whenever we're talking about oxygen, why do we need it? It's aerobic respiration. In our key terms scan, doesn't hurt to have that in there. You can say for nutrients, obviously, if you can give an example, e.g. glucose for aerobic respiration. And then all of these are 1 marking point. So this is this one, or this one, or this one. So sufficient removal of a named waste product, or for aerobic respiration or metabolic demands is one thing we can say there. So if you said oxygen for aerobic respiration, that's 1 mark, not 2 marks, because this is an or. Okay, no, actually, it's not totally clear whether that's the case, or actually maybe, I'm just going to zoom in a bit here so we can, what is this or for? So I think actually it's probably this or that. So if I number these, let's number them all. Ugh. So I think what this is saying here is we've got marking point 1, marking point 2, marking point 2, and marking point 3. So this is either or, either delivery or removal. Yeah. In fact, that's not right either. My numbers are wrong. It's amazing how concentration goes after, and then it's sort of 3A or 3B, and then marking point 4. That's more like it, I think. Lucy finally got full marks. Okay, so okay, so I'm going to throw that poll question at you again, the same poll question. And I want to know, just for this question, the 3 marks, where did we lose marks? Looking at the mark scheme, do you know all of these points? Maybe you do. And if you know all these points, then it can't be content knowledge that helps us. If you don't know that larger organisms have a smaller surface area to volume ratio, then that's a content point that we don't know. Vanessa, I thought I missed a question from you just before. Yeah, always chucking in exampling. Always good. Oxygen for aerobic respiration, glucose for aerobic respiration, whatever. Sodium for name a role of a sodium ion might be, well, if you do A level, then we're talking about got nerves, action potentials. So how do we know we need to include the reason? Because it's an explain question. command word. So if you didn't give the reason, someone's saying on YouTube saying that they didn't give any of the reasons, well, that's question analysis for me. That's because we didn't pull the command word and say give reasons for. So in my head, I would have said give reasons for why the surface area to volume ratio determines the need for circulatory system. Okay, so if it's any of, basically, if it's these marking points, if you didn't know, obviously, if you didn't know that they're the reasons, then it's content. If you do know, when you're looking at these, oh, of course I know that that's the case, or I do know that it's to remove waste products, or I do know that it's for aerobic respiration, then it's a question analysis point. Okay, so I'm going to end the poll again, share the results. This time, 72% of people drop marks for answering technique. And that's the biggest one. We had 11% for question analysis, 17% for silly mistake, and 20. So less than 25%. Less than 25% of the dropped marks are for content. So if you're spending your entire revision working on content, you're probably only focusing on improving the 25% of your marks. So what I'm trying to do is like bring our awareness to the fact there's lots of other things to revise that give us more bang for our buck than simply churning through content and improving our content knowledge from maybe like whatever, arbitrarily I'm at 60% content knowledge, I might get to 70% content knowledge, but that will translate into some marks on the exam, but not lots of marks. Okay. Does anyone remember what your homework is for today? You may not have joined the beginning of the live. I stated at the beginning. Homework to all of you today is to find one application question. One application question and submit it to me, probably on the live classes course. Let me actually, I've got one of them open here. For once, I'm going to demonstrate with an exam board that is an AQA, but it doesn't matter which one we choose here. Let's go with, that's not what I want to do. Let's do this. Okay, so from the live class page, I will update both of these links. I think where students were saying that the link wasn't working is that you were using the link for last week's class, which was here, and I should have changed it. It was my fault. So make sure you're using this link, but I think I've updated them all now. So if you just ask me a question down here and post, you can attach a screenshot right here with the question, or else you can give me the full question code. So I need to know the exam board. I need to know the paper number. Is it an AS paper or an A-level paper? The year and the question number. If you want to just send it's Rich, it's EdXL AS paper 2 from 2016, question 12. And then we're going to do some more application questions tomorrow. I'm going to talk you through how I would approach them, where the clues in the questions are, and how we can like hopefully get stronger. Obviously, to get good at application questions, we have to have understood most of the content, because it's going to sort of rely on you understanding content piece A, understanding content piece B, and then often it's going to join those two together, which is again all the reason why only doing single topic questions and topic question pack, topic question pack, topic question pack, often you don't get those two topics being joined like this. And if you're not practicing those styles of questions, you're going to get it for the first time in the exam, and it's going to be much harder for you to get the marks. So that's why revising paper by paper using the TT revision method is the best holistic approach. Okay, so I'm going to just make sure I've got the chats open. I can see any questions are being asked. I'll come back to YouTube as well. Yeah, on YouTube, can you say the opposite? A smaller surface area to volume ratio, more easy to diffuse. Well, that you can say it the other way around, but what you've said is not correct. Smaller organisms have a larger surface area to volume ratio, and so it's easier for substances to diffuse. Is that what you're saying? Yes, you can basically use or the reverse argument in any question. Okay, Lucy. Yeah, I mean, and next week, this week, 5:30, 5:30, 5:30, Tuesday, Wednesday, Thursday. Next week, back to the usual one session each week at 5:30 on a Tuesday. And I'll be doing my sections of AS and skills and A level. I don't know, I haven't even thought about next week's live classes at this point, but I will keep the page updated as we go through the week. Can anyone remember any specific application questions? Or you don't have to tell me the code, but like what the topic was on and the exam board would help, that you got stuck on or have got stuck on in the past, or maybe you succeeded in doing it, but you were very proud of yourself for doing so. So yeah, let me know. If you're on YouTube, you can obviously contribute with that as well. Just let me know, oh, there's a question from OCR that was about whatever. I know the papers well enough that I can probably find it. Raizen in YouTube, feeling more confident application questions. Thank you. My pleasure. Well, we're getting there. AQA action potentials. Can you give me any more than that? That will definitely be a starting point. Give me some key terms that will either be in the question or the answer is another way of doing it, because I can then do a search through that. Was it about yeah, were there any? Yeah, perfect. If you can find it, then just take a screenshot and send it through to me and I'll be able to prepare it. Marima, long passage Alzheimer's one. I think that's AQA, right? But it could be, I'm pretty sure OCR, I haven't done that. Marima, okay. Was that, Marima, just let me know the exam board for that. Okay, excellent. Yeah, I think I know the one you're talking about. Block passage of text. It's about gene regulation. It's probably mostly A-level content, so I might have to stick that one at the end. If you can find an AS ones, then it will be great, because I'm going to do at least the first half on AS questions tomorrow so that AS students can join us. Question 2.2, 2018 paper on surface area and osmosis. Again, Fatima, let me know the exam board, and is it presumably an A-level paper? Probably the best place to upload the questions is onto the live classes page, in the comments on the live classes page, and just say, hey, Rich, this is for tomorrow's live class, and I'll just check that they're suitable. Some questions are like, don't fit with some exam boards, and I'm trying to make these inclusive. So I might just choose not to include some. Yeah, we can maybe do some marking. Can we mark some people's responses to exam questions and discuss whether... Yeah, what? Okay, so basically how to mark. Basically, that's your homework. Spend 5 minutes, find one question that's an application question, send it through to me. I'll then have a nice selection that I can pick some good examples for tomorrow. And I'm going to basically, okay, yeah, take a shot of that. I might pull in some pre-answered questions, and maybe we can look at what they've done wrong and maybe some ways that we could avoid making those mistakes. Often their technique is not going to be right. Okay, wonderful. Well, I think that's everything from me. I will see you same time, same place tomorrow. And Berry, you had a question for me earlier on that was about unrelated to the live class. So ask me in the comments on the course. I kept an eye on the Q&A, but it doesn't look like there was a question that arrived in the Q&A. Oh, there was one in there. So my bad. I had it covered up. I'm just going to read it in my head beforehand. Okay, so this question is something that I'm going to answer this live because it's going to be the fastest way Barry's going to get an answer. It's quite applicable to a lot of people. You feel like you're running out of time to revise in depth for your tests coming up. You have a paper next Tuesday. In fact, maybe the chat can answer you here because you might be able to predict what my answer is going to be. You have paper 1 next Tuesday and paper 2 next Friday. What's the best way to revise for these papers? You don't feel like you have enough time to answer past paper questions separately on each topic. How many hours should I do a day? Okay, well, obviously time is running out, But that doesn't mean that we have to go into a crazy panic of a tailspin. Honestly, Barry, I don't know any way that's more effective than using the TT revision method. Well, you've got paper 1 and paper 2, so find out exactly which topics are on there. If you know you've got an absolute shock horror topic, then maybe go and patch up that topic just quickly before you start TT revision method. But do at least one cycle of the TT revision method on a paper 1 and one cycle of a TT revision method on a paper 2 before next Friday. That's totally achievable. Trying to achieve more than that might stress you out a little bit. Genuinely, making sure you are focusing not just on the content that you get wrong, but if it is, I'm not understanding the question, go back to the exam guide, make sure you've watched the answering technique. You can watch the live classes from last week. You've also got the revision technique in there, which are quite short videos on like how to make sure you're making the most of your time. During Easter holidays, you might want to be putting in, I'd say 6 action hours, 2 action hours per subject per day is when you're not at school or college. Maybe extend that to a little bit more, but I'd say 2 action hours per subject per day. And then when you're at school or college, you want to be maybe doing per week 4 hours of lessons, 4 hours of homework, and 4 hours of additional revision. So that's 12 hours per subject per week when you're at school or college, and then probably 2 hours per subject per day, 5 days a week when you're not at school or college. I'm not doing any A-level content today. No, this week I'm doing 6, well, last week and this week I'm doing 6 live classes focused around exam techniques and skills. So I'm going to return to the normal format. I might do some A-level tomorrow. It depends on which questions get submitted to me. I'm going to do at least the first 35, 40 minutes on AS. Then I might tack on some A-level at the end if I think there's a really good question. Okay, thanks, Barry. Crack on the TT revision method. If you get stuck at any point, then just reach out to me in the course. You know where I am. Bye.